Alright students, hello and welcome to AC24. This is our taster gene, or we're trying to identify a modified gene in a population. What we're going to do here in this lab is we're going to use PCR, restriction enzymes, and gel electrophoresis to try to determine the genotype of what we call the taster gene. It allows you to taste a bitter food. Some people have the taster gene and some do not. It's a single base pair mutation that allows you to taste or not taste. So that's pretty interesting. What you're going to do is you're going to do a little pre-lab. You're going to take some of this PTC paper, and if you do have the gene for bitterness, you will taste the bitterness on the PTC paper. And if you're homozygous recessive, you will not taste the bitter paper. So here's homozygous recessive, you do not taste it. If you are homozygous dominant, you will taste it, and in fact you will taste it very, very strongly. You probably won't like it very much. And if you're heterozygous, you might taste it just a little bit. You'll probably be able to taste the bitterness, but it won't be very, very strong. Okay. Remember, these are different alleles of the gene. You get one from mom and one from dad. So you should be able to possibly figure out what your parental genotype is based on what you are. That's pretty interesting. So what are we going to do, right? We're going to, step one, we're going to swab our cheeks with a sterile loop here. We're trying to get our own DNA out of our cheek cells. So it does need to be fairly vigorous. You do want to get a lot of DNA. This lab doesn't work quite as well if you don't get a lot of DNA. So you want to be fairly vigorous when you're swabbing your cheek, but of course you don't need to draw blood or anything like that. It doesn't need to be super, super strong. Then what you're going to do is you're going to take your loop and you're going to dip it and twirl it into the microfuge tube that you labeled that has this Chelex solution in it. Alright, and so what Chelex does is it is it's a little guy, and what it does is it binds specifically magnesium, it binds ions, and nucleases, which are inside of your cells. They naturally break down DNA, or DNA aces, I guess they're also called. They break down some of the DNA naturally inside of your cell, and since we want the DNA whole, we want to look at it and amplify the certain sections, we don't want that to happen. So what this Chelex will do is it will bind to magnesium, and that will allow the DNA aces, remember it's an enzyme, it's got ACE. So the lack of magnesium, because the magnesium is bound by the Chelex, the DNA ACE will not function and it will not break down our DNA. And our DNA will be whole, which is what we want. Alright, so that's the function of Chelex. We're also going to add a solution, 2 microliters, of something called proteinase K. And as the name suggests, ACE, so it is an enzyme, protein, breaks down. Alright, so this enzyme breaks down protein, and it's going to break down any excess protein that's in our cells. Break down protein. So it will help to clean up, so to speak, the DNA. We want nice pure DNA. We don't want any excess protein floating around in our cells and that sort of thing. So we're going to add the proteinase K. We're going to add DNA or cheek cells. And then we're also going to add this Chelex solution. That's all going to be in a tube. And we're going to put it into the digital dry bath for 20 minutes at 56 degrees. Why 56? Well, right around 50 degrees or so is the optimum temperature for the proteinase K to work. So we're going to let that stuff go to work for about 20 minutes, and then we're going to flick 10 times. Centrifuge to try to get all of the solution down to the bottom of the tube. You'll notice we're not centrifuging for very long. All we want to do is get any of the liquid that might have come up on the walls down to the very, very bottom, because then we're going to boil the water in the water bath for 8 minutes. And what we're trying to do here is denature all of the enzymes that we've added to the lab, right? We don't want those enzymes to keep going. We want everything, once it's broken down, to sort of stop and be ready to go. So then we're going to mix and flick our tube again. We're going to microcentrifuge for about 3 minutes. And again, we want to get all of this stuff down at the bottom, right? So all the protein that the proteinase K has digested, all of the cell membranes, all the cell junk, basically, right? So Here's our little microcentrifuge tube with a cap. It's the worst drawn microcentrifuge tube ever, I realize. So we've got this microcentrifuge tube and we've got a little liquid here. Right? We want all of this junky DNA membrane lipid stuff down at the bottom. So our DNA will hopefully, oops, sorry about that. Our DNA will hopefully be suspended up here in, oh my gosh, it will be hopefully suspended up here in the supernatant. And we can grab it out of there. Okay, so that's why this step, it's very important to be gentle and not mix. You do not want to mix up your DNA with all of that junk. Okay, so we're going to label a new 0.2 microcentrifuge tube, that's the very small tubes, and label it PCR Taster. Into that, we're going to pipette 43 microliters of our PCR master mix. This is the same master mix that we used last time, right? It's the same mix of stuff, so you're going to have things like ATP, nucleotides, buffer, and primers. Okay, so primers, even though the word is the same, primer, as the last lab, AC23, they're not the same primer, right? In the last lab, in AC23, we were trying to amplify the gap DH gene. Okay, so we needed specific primers for that we put upstream and let them work. In this lab, however, right, we're trying to amplify the taster gene. So the same primers will not work. We need a different primer upstream to allow the PCR to take place. So it's still the same word, but it's different actual primers, right? They're going to be a different combination of nucleotides in this one. So we're going to add the PCR master mix to our tube. We're going to take some of our DNA from up here, right, and add that to the tube as well in this step. So now we've got everything to run the PCR reaction, including, hopefully, our exposed DNA. And our primers will select just for the taster gene that we want. And when we run it in the PCR machine, right, it will hopefully make millions and millions and millions of copies of just our taster gene, which is what we're looking for. Here is our PCR cycle. It's fairly similar to all of the other cycles, right? Notice we have steps 1, 2, and 3 here, and then it's just going to hold at 8 degrees forever, and the DNA will be very stable. So over here I have a quick little picture of PCR. Right, here's our very first step down here. The DNA is double-stranded, just like normal DNA is found. What we do is we increase the temperature, and this denatures the DNA. I know we've talked about this word before. So denaturation in this case means that the DNA will split and spread apart, and so you'll have single-stranded DNA. And then what you do is you cool the temperature down, right? And the primers that we put in over there will anneal. Another word for anneal, we could use bind, right? We could use attach. We could use any of these words there, but annealing is the technical term. So the primer that we use will anneal or bind to the DNA. And then you heat up the PCR machine again, which allows the polymerase to extend that new section of DNA in both directions. Oh, wow, look, I forgot something, sorry. In your tube, you're also going to have the very important thing, right? You're going to have the Taq DNA polymerase, which is the enzyme that will actually make the new strand of DNA. Almost forgot that one. He's very important, though. Right? That will be the enzyme that actually does the copying here, back and forth. Very important enzyme. All right, and so that will allow you to hold your tubes at 8 degrees, and then you can go on to day 2.